Is this a game changer? If we can get to that point where we can really revolutionize cancer care and kind of improve the accuracy, improve the turnaround times, that is a game changer for us.
So I am Torbjörn Kronander. I'm the CEO of Sectra, providing the packs, so the imaging packs, for both pathology. We can also do radiology for Genomics England.
Thanks, Torbjörn. So my name's Dr. Prabhu Arumugam. By background, I'm a pathologist, and I am the Director of Clinical Data and Imaging at Genomics England, and currently head up the multimodal programme.
The term game changer is very often used. Is bringing data together like this a generally game changer, do you think?
I think bringing it together is setting a route that has not been done before at this scale. I think the game-changing bit will be the analysis. So just to put this in context, there have been lots of attempts to bring together multiple sources of data or multimodality. And there are lots of kind of what you might call databases or libraries of pathology images on their own. And there's huge examples, not only in England, but in America, of kind of tissue-specific or disease-specific pathology archives. We have lots of publicly accessible radiology archives. But to bring that all together, the previous attempt was by the NIH, and they had the TCJ Atlas. So they had a couple of thousand genomes, about 20,000 pathology images. And if you think about it now, most AI or cancer kind of based diagnostic machine learning algorithms and programs have run through TCJ. And that's been the real success for kind of a lot of organizations the last kind of 10 years or so. But to put this in context, what we would put together by the end of this would be 17,000 cancer genomes, about 250,000 pathology images, and probably an equal number of radiology scans. So the numbers that we'll be getting is far outweighs what TCGA has, but then we'd also be attaching that to a very high quality clinical data set. So we have outcomes from the National Cancer Register here in England, including histology, what kind of chemotherapy they've had, or what we call a SAC data set. So there's a vast amount of clinical data that we can also attach to that, including kind of unstructured pathology reports as well. So that asset in terms of what we can bring together and the way we try to push that into really forward-thinking ways. So deploying this all in the cloud, making it all available through researchers from a cloud-based front end has been really novel and really interesting. And that's the bit that's revolutionary. I think the game-changing bit was how then that is going to impact patients ultimately, because that's why we're doing this. It's how do we make those diagnoses better? How do we make the outcomes for those patients better? And we're just trying to push the boundaries. But I think what I would say is that it's how our researchers and our commercial partners and industry partners use that data to make those diagnoses. I think that's going to be the game changer.
You're basically a research-oriented organization. What do you think this could lead to for change how research is carried out? I mean, you will have one of the largest data sets on the planet.
Yeah, that's the aim. So I think there's multiple kind of ways to break that down. So we like to lead research. We like to make sure that other researchers can also answer their questions. If I was to answer all the questions I wanted to do just on the data set myself, I'd be very skewed and very focused on what I was interested in, which is unfair. This is such a unique data set. The whole point for us is to make sure that everyone who has a valid research question can access that data. And ultimately, the important bit here is that it's anonymized or pseudo-anonymized. So they don't know who the patients are. They will know, for instance, this is a 50-year-old female with breast cancer that has, had a recurrence 3 years later, and that's all they will know, and that's the important bit. So the whole point and the importance for us is to make sure that other researchers can access that. And at the moment, we have two kind of front ends to access our data set. So you're either a clinician or an academic, and we kind of loosely term that GSIP, or Genomics England Clinical Interpretation Partnership. Or you might be a commercial partner. And I'm really open to the idea of not distinguishing those two, because actually both organizations, both groups of people have very interesting ideas, and we should be in a position to support both. But we do distinguish that ultimately, if you're a startup, we do provide you free access, and the same for academics. And that's a really important bit, that, we don't want to charge you to access this data set, because they will all have great research questions that we have to support. And that's the important bit, is that we will never be able to answer on our own every kind of potential or theoretical kind of research or clinical question. We just need to make sure that we're providing the tools, providing the workspace and the environment for others to answer theirs.
And do you think we can change who does research through this initiative? I mean, there will be a lot of IT people merging into genomics now.
Yeah, so I think that's a really interesting thing that we've also faced as well. So everyone talks about who's your end user and how you're going to integrate with your end user. So historically, we as Genomics England have always engaged with bioinformatics or bioinformaticians. So people that really understand the genome, how the genome works, and actually their questions are very heavily focused on what they understand. But actually, this data set really does pivot everything around. Actually, we're going to go from academics to clinicians. So pathologists will want to start using this as data. They want to start annotating the images. Radiologists, so actually, can you start correlating what radiologists are seeing on like MRIs and CT scans? Are there things of interest that actually they've also started to advise? So these are already new groups of users that we're going to start engaging with, and actually how they interact with our data set is also then very different. Like actually what you want is someone to be able to engage with like a point and click interface. So we're really pivoting on how broad our user kind of database is going to be, and our their knowledge and skill set is going to be. And it's brilliant. I think, if you can bring in clinicians in particular, and clinicians I use very loosely, but, that could also be kind of cancer nurse specialists, because they could also kind of interpret what they see at an MDT level. If you can integrate kind of clinical genomic and scientists, there's lots of people in that kind of clinical MDT that this data set could be really accessible and really tangential to. So I think it's just trying to make sure that they understand that there is a use case for this as well, and how we can bring them into that fold very early on as well.
And how do you think it will help to inform new drug discoveries?
Oh, yeah, I think that the doors here are potentially vast in terms of where this data set could go. You could almost break it down into which kind of pathway you want to go. So do you want to kind of correlate morphological changes? So you might say, actually, these changes we see morphologically, or kind of a digital pathology image, that wrinkling of that nuclei probably actually correlates to a BRCA1 mutation. We don't know. Or are we seeing something that correlates to maybe a HER2 change? So the retrospective analysis in terms of how you could make those better diagnosis morphologically is one pathway. How you then correlate all those individuals to making better decisions. So, should that person, based on what you've seen from their genome and from their kind of imaging, actually they should have neoadjuvant chemotherapy first, then have an operation. Again, those kind of precision medicines and precision focus is great, and it's another avenue. And then the other bit is novel discovery. So targeted drug discovery is, everyone talks about it, but actually what are you looking to do and how are you going to do that? So the targets in the genome, I think, are then the bits that from all of the clinical and the imaging that we have, how we can then map that back to the genome, I think is going to be really interesting and really revolutionary. But I think for us to work through that just now, I think we're probably at the very beginning of that journey. So you're probably going to be seeing some really interesting stuff probably in the next couple of years. And I think that could be really interesting, even like just repurposing kind of other drugs for diseases that we already know about. Those are kind of things that we think we've done. Pharma spends billions and billions of dollars on kind of creating new drugs, and some don't work. But actually, if you could refashion them and use them for other diseases that we hadn't really thought about, that's really interesting. And actually, I think that puts us in a really good position in terms of, costs of treatments and better treatments ultimately for patients.
So let's go into little clinical aspects here. What do you think Genomics England will mean for future care? Connection of combined data. Do you think we will run AI over multi-omics in the future, for instance, and not only pathology or radiology?
So the aim that we're trying to kind of direct England to per se is actually where you do whole genome sequencing, you could also generate a digital pathology image from that fresh frozen tissue. So the focus there is how to get a better, quicker, more accurate diagnosis back to that patient in real time. Ultimately, that's aim number one. But then, the opportunities here are actually really interesting. So if you can start correlating that all together, can we use AI to make those diagnoses even more accurate? So can we say, actually with this patient with these features that we've identified, the correct course of treatment should be this, they should have this operation, and, their 5-year survival is going to be 82%. Can we get to that kind of level of accuracy? I think that's where AI is going to really be key, because actually, like I say, the more relevant bit is AI is the one thing that can parse through all of that data. You and I are not going to sit in front of like a screen and look at every genome and kind of every codon potentially known to man, but an AI system could, and that's relevant. I think the decision support part of AI is the most tangential thing, and I think that's what clinicians will find most relevant. Actually them saying that this is really kind of significant, this is how is our suggestion from a model, and this is how you should be treating individuals, or this type of treatment they should be having. I think that is something that, is what's been wanting and been calling out for cancer for so long. And I think that's something that we can help as Genomics England get to that point in the next, say, 3 to 5 years. I think that would be really interesting. And I think, going back to your original point, is this a game changer? Right now, this data set isn't, but if we can get to that point where we can really revolutionize cancer care and kind of improve the accuracy, improve the turnaround times, that is a game changer for us.
What is significance of doing this in a cloud environment? I mean, of course, 500 petabytes would be kind of a little expensive doing it on-prem. But what are the benefits doing things in cloud? And we have also helped you with the images in the cloud here.
If you're all in the cloud, the ability to link data across TREs or secure data environments or some kind of cloud-based storage is much more appreciable than, say, if you're on-prem. And that's the problem, is that a lot of incredibly valuable data sources are stored basically in some hard drives, in some research institute that no one else sees. And that's great. Like, they've got a very secure way of hosting that data for themselves. That's fine. But what I wanted to showcase is that what is possible in the context of being deployed in the cloud. So it's not only the storage costs, it's also our cloud providers provide us a huge amount of security. We're incredibly kind of comfortable in the notion that we can host identifiable data, obviously only for specific kind of Genomics England employees for reasons of kind of linkage to other data sources, but we hold identifiable data. The images, for instance, that come over IEP are from NHS sites, so they are identifiable data sources that we retrieve into our cloud storage, but then obviously then anonymized in an ongoing process. So the security that also provides to us is exceptional. There's duplication of data, so if anything does happen, we're not going to lose anything. But then there's also the kind of the theory of deploying tools, running analysis, how we make kind of users from around the world and how we make that much more accessible. I think those are the kind of markers I was trying to drive towards. But then also the longer term vision is that actually kind of cloud storage, digital pathology, that all links very nicely to actually, if you're an expert in a very specific cancer in Australia, you might be asked to kind of provide support and input into like really unique cases, say for instance in England, and actually that collaboration, that better diagnosis does also come from expert input, and maybe that only comes from kind of having that forward thinking approach, digital pathology that's in the cloud that you can access from anywhere in the world. So I was just trying to showcase what is possible. And don't get me wrong, there have been extraordinary challenges. It is, yes, beyond the size of the data set, deploying kind of de-identification protocols, making sure that users access that data set in the right way, building out our kind of data lake, linkage tables. It's all been hard, and we've had to build a team with the experience and the knowledge to do that. But if we can't do that, if we can't showcase the value, I think we'd be doing ourselves a disservice. So it was very much a forward-thinking project. We've learned a lot, and we want to continue sharing our learnings to everyone else, really.
You mentioned data security, cybersecurity, which I agree completely is better than in the cloud and on-prem. And I think the UK realized that after WannaCry. I mean, the UK changed completely after WannaCry discovered that patching of servers on your own environment might be more difficult doing it in the cloud. But apart from that, how do you see, DNA is by default, identifiable. That's the idea of it, right? But you can make it reasonably difficult. How else do you see cybersecurity questions being raised and addressed in this project? Now, and how have we helped you out in doing that?
Okay, so we'll start with Sectra and your kind of really important bit to us. So going back, we ingest images with identifiable data in them. So that includes patient names, NHS numbers, sites, and they're all in the DICOM tag. And that is a vast amount of information. Actually, you will also get hidden in that as also the radiology report. So the report's hidden, or not hidden, but a part of the transfer. So this is all very identifiable information. So you can classify it according to the ICO as identifiable information or sensitive information. And slightly strangely, genomic data is actually sensitive rather than identifiable. So the important bit here is we have a very strict protocol of what we define as identifiable, and we need to make sure that for researchers, that was not going to be kind of made available. So all of our participants have what we call a GEL ID or Genomics England identifier. And that could be the only thing that you can track across. So you should be able to build a cohort based on your participant ID. So the images, both pathology and radiology, will have the same ID or the GEL IDs, as does the data set, clinical data, as does the genomes. And Sectra are incredibly organized in the way that they helped us build the anonymization of the DICOM tags. So at the moment now, what we have is two environments. So the identifiable data lands, and then they run through an anonymization process, which then literally leaves us with the same scans or duplicates of scans with only our GEL ID and ultimately what we wanted, which is just an age and the data scan. And you can imagine how the amount of data which is taken away from that then just allows those images to be used completely anonymized in the research environment. And that has been game changing for us. That is revolutionary in the way that, we don't need to do that manually. We're really comfortable with the way that Sectra set that up for us. And is a real kind of, for us, has been unique because actually we can do that all in the cloud, all in one go. And actually for hundreds of thousands of images, that's a fascinating way to kind of help us allow researchers quicker access to that data to move on. So one is what you guys provided, which I think has been really instrumental for us as GEL and for how we worked with participant and patient data. The other bit for us is obviously is beyond that is, yes, the genome is potentially to some extent identifiable, but we have very strict kind of procedures in terms of who we allow in. So anyone who wants to apply needs to have write an application for the kind of research they want to do. And also we check in terms of what their kind of reasons for doing that is. And then also they have to do a governance kind of test as they kind of enter and onboard. So it's very clear that they have certain rules that they need to operate within. But then also you can't take any data out of our environment without a specific kind of reason. So what we say is that you can take out kind of images or large scale analysis. So you might say, for instance, you want to look at the number of participants that have had left hemicolectomies with, I don't know, like FOLFIRINOX as a chemotherapy and have an MSI high kind of cancer, and that would just be a numerical count. So we're quite comfortable with those to be released, but beyond that, you can't take anything out of our environment. So you have a process called airlock, which is, I think a lot of people refer to it as being quite strict and quite militant, which I agree with. That I think that's a right process to have. But it reviews quite a lot of these edge cases in terms of what is right to release. We don't release row level data. It's only aggregate numerical counts or summary kind of pictures for publication purposes. So that provides us to control also ourselves in terms of vetting who comes in and also vetting what goes out, so that we're very much in control. Because otherwise, the data that we hold is vast, it's huge, and you could spend a lot of time exploring that in its kind of in all its complexity. But the important bit is, we want to know what you're taking away. But then the kind of also the other push we also have as research is also start returning your interesting findings back to us, so we can start telling our participants and our participant panel what has been found, what we're using their data for. And I think that is probably the key to me, is that the data set we hold is not really ours, it's our participants. They've provided that to us, that's their kind of donation to us, and we're in control of their data. So we are their guardians, and we've got to protect their data in whatever way we can in the right way, but equally be open to what is useful for research and how do we advocate better diagnosis and so on. So it's a fine balance.
Assume you, in a very far distant future, finally decide to retire. What would make you really happy and proud of the outcome of Genomics England?
So to be honest, Torbjörn, I think what we've done thus far, I'm already proud of. We've really kind of pushed the boundaries of what is possible at the moment, and I think the team that we've built, the progress that we've made in the last couple of years has been brilliant. And if I was to die tomorrow, Torbjörn, I'd be proud of what we've done. I think the bit that I would like to really advocate is that looking forward, that this data set is actually used. I think the bit that I'd probably turn in my grave is it was kind of locked away and kind of it got locked in terms of some kind of, I don't know, governance issues or something like that, and it never got used. I think that would be the biggest kind of thing that would really upset me. I just want to make sure that this is used, but used in the right way. And as long as I know that we're impacting patients directly, and also clinicians, that we can help them make better diagnoses, we can help support them in the kind of the difficult and complex diagnoses, and that it's impacting patients, so it's giving them quicker results, better results, more accurate results, then I'll die a happy man.
What's your advice for Sectra going forward? What should we do next?
I mean, the world is your oyster, isn't it, Torbjörn? So I think, going back here, the reason we wanted to work with Sectra is your willingness to, operate across multiple cloud environments, your willingness to kind of host multiple sources of data, your willingness to host other kind of third-party apps. And that's really unique. So I think your willingness to kind of work beyond clinical systems is great. And I think that's what kind of attracted us kind of very early on. And I think that's the bit that probably Sectra will continue to be unique, is actually your openness to new technology, new diagnosis, and not only just say, yeah, it's there, we don't really care about it, but to actually start integrating that and have the vision and the foresight. That's really kind of where Sectra's got to continue ploughing on, because if you can be that one source of, this is a program that all NHS sites or all clinical sites need to use, and it can host all data sources. Ultimately, how we use that, I think, is still open. But it becomes our warehouse, it becomes that kind of linkage and everything to us. It's just much easier, and I think that's a bit that clinicians in particular could really engage with, and that's where that value as an organisation will be unique to you guys.
Yeah, we also see, I mean, we had a discussion about tumor boards or the MDT for cancer, and that's a very, very expensive piece of healthcare because it sits a lot of very expensive doctors there. So our vision is also forward, provide all the data in an integrated fashion so we can speed up the tumor boards and thus diagnosis. That's what we are. So we're very proud of working with you. We wish you all of luck, and we will be there helping out whenever you want. And with that, I would like to thank you very much for your time in the interview here.
No, thank you very much for having us. And yeah, we'll obviously keep in touch, keep you updated about the progress, and look forward to continuing our relationship together.